Okay, guys, today we're going to talk about section 12.2, which is the replication of DNA. So let's go ahead and get started with that. The main idea of this section is that DNA replicates by making a strand that is complementary to each original strand. So essentially, we are going to start with a parental strand of DNA. Remember, DNA is double helix, so there's two strands. So we're going to start with that parental DNA, which is a double helix, and we're going to replicate that and make a new strand complementary to each original strand. So let's talk a little bit more about that in detail. First, DNA replication is semi-conservative, meaning that the parental strands of DNA separate, and each individual strand then serves as templates and produces DNA molecules that have one strand of parental DNA and one strand of new DNA. So there are three main stages. The first stage is unwinding. The second stage is base pairing. And the third stage is joining. So here's a picture just to kind of break that down a little bit. Here on the left, we have the original, we have that parent strand. Then we go through the first replication and notice that each one has two strands, but each one has one of the original strands and then one new strand. So that would be the first replication. Then if you go on to the second replication, well then only one of those offspring are going to have a piece of the very original parent strand. So talking about step one specifically, the unwinding or the unzipping phase, DNA helicase is an enzyme responsible for unwinding and unzipping the double helix. So that unzipping leads to those hydrogen bonds between those base pairs being broken, which leads to the single strand of DNA. So it literally is like a zipper to where, like if you have your jacket zipped up, it looks like one, but as you unzip it, it separates into two separate pieces. And that's what happens with DNA replication, is that DNA helicase acts as that zipper and it unzips that double bond or that double helix and makes it two single strands of DNA. So then RNA primase, that's another enzyme, and it adds short segments of RNA, known as RNA primers, onto each DNA primase, which we'll talk more about that later. But the replication fork is the point at which the DNA unzips. So as you go through DNA replication, you'll see pieces of the original DNA to where there's still a double helix, there's still two strands. The replication fork is that point. It's literally like a fork in the road, like where you split off into two different strands. So here's just like a little GIF showing you that's exactly how DNA replication works. So you have them connected, then you have the DNA polymerase coming in, and the DNA helicase coming in and unzipping that double strand. So once it has been unzipped, we move into step two, which is base pairing. So DNA polymerase is an enzyme that catalyzes the addition of appropriate nucleotides to the new DNA strand. So remember, an enzyme is something that speeds up the process, right? So this DNA polymerase comes in, and now instead of one original DNA that's a double helix, we have two single strands of DNA. And what's going to happen is this DNA polymerase is going to come in and it's going to add nucleotides to each single strand. So remember when we talked about Chargaff's rule, adenine can only bind to thymine, so A to T, and cytosine can only bind with guanine, so C and G. And so that's exactly what happens. You have these strands, these single strands of DNA, and they have either adenine, thymine, guanine and cytosine on that original strand, and then the complementary strand is built, and it DNA polymerase adds the complementary nucleotide. So, for example, we're going to talk more about that. I know that sounds kind of confusing, but it's important that you know that nucleotides are added to the 3' end of the new DNA strand, meaning it's complementing the 5' end of the old strand. So you're going to have two ends. You're going to hear it referred to as 5' and 3'. Okay, so here's what is kind of tricky. Nucleotides are always added to the 3' end of the new strand, but the old strand, it's complementing the 5' end. So look at this. If this is our original strand, this is after it's been unzipped, okay? This is our original strand of 5'. All right, and we have adenine, guanine, cytosine, thymine, adenine, guanine, cytosine, 3' end. So that's like the end of that segment. So this told us that nucleotides are added to the 3' end of the new DNA strand, complementing the 5' end of the old strand. So this is our new strand here, and it is going to read literally the opposite of what this did. Kind of like how I told you before that it's like a magnet, right? Like positive and negative are always matched up together. Like when you put in a battery, you have, if it's two batteries, one of them has the negative end facing up and the other has the positive end. The same is true with this. So the 5' end of the original strand is going to be complemented by the 3' strand of the 3' end of the new strand. That's how we get that anti-parallel look. But they complement each other. So remember with Chargaff's rule, with adenine, the complementary base pair of adenine is thymine, right? So everywhere you see an adenine on the original strand, there's going to be a thymine on the new strand. The same is true with guanine. The complementary base pair of that is cytosine. So everywhere you see a guanine in the original strand, you're going to see a cytosine in the new strand. So there are the two strands, right? There's the new pieces. There's a leading strand and there's a lagging strand. So the leading strand is elongated as DNA unwinds, and it's built continuously by the addition of nucleotides to the 3' end. So this is an example of the leading strand, okay? Because this is our original, and the new strand is just added really simply by complementing the 5' end and adding to the 3' end. However, with the lagging strand, it elongates away from the replication fork. So instead of going really easily in this direction, it kind of has to work backwards. And because it has to work backwards, and because that's not necessarily the natural way for it to happen, it's synthesized discontinuously into small fragments called Okazaki fragments. So here's a picture. If you notice here, this is our parental DNA. So here's our 5' strand, and here's our 3' strand. Now notice this is the leading strand because, as you can see here, we have our 5' end, okay, and things were added in the direction in which that it should have been added in. Okay, things were added 3' to 5'. But here it's working in the opposite direction. So it's a lagging strand. And so we've had to form these small sections, which are called those Okazaki fragments. We've had to do it a little bit at a time because it's not the natural way per se. So then finally, we have step 3, which is the joining. So once DNA polymerase comes to the RNA primer, which is this right here, the RNA primer has to be added first so these Okazaki fragments can be added. So DNA polymerase will come to those RNA primers once Okazaki fragments have been added and will remove them and replace them with DNA nucleotides, which is like the adenine, thymine, all of those things. So then once that happens, DNA ligase will link the sections together once that RNA primer has been replaced with the nucleotides. So it's kind of just like the seal, right? DNA ligase just seals everything together. So to kind of break this down in a little simpler form, we first have helicase. Okay, so helicase is that enzyme that breaks down hydrogen bonds. And that's what causes the DNA to unzip. So remember, it's hydrogen bonds that have the base pairs bound together, like adenine and thymine are bound together by a hydrogen bond. Well, helicase comes in and breaks that hydrogen bond, causing the DNA to unzip. Then it leads to that replication fork, which is the point at which the DNA unzips. Once it's unzipped, DNA polymerase comes in, and that's the enzyme that adds nucleotides to make the DNA. So that's where we had the 5' end and the 3' end, and they were complementing each other based on Chargaff's rule. And that DNA polymerase proofreads and makes corrections. So it just kind of scans and makes sure, oh, yeah, A and T are lined up, C and G are lined up, and vice versa. It doesn't want to pass on any mistakes because that ultimately leads to damaged DNA, right? Which we'll talk more about that later. So here's just kind of a summary of the enzymes in DNA replication. We have the helicase, right? We've said that several times. That's what unwinds and separates double-stranded DNA as it moves along the DNA. And that's what forms the replication fork. Then you have DNA primase. That's a type of RNA polymerase that generates RNA primers. And RNA or primers are short RNA molecules that act as templates for the starting point of DNA replication. Remember, that is what is needed. Those have to be there before the Okazaki fragments can be added in. So then DNA polymerase is what synthesizes those new DNA molecules by adding nucleotides to leading and lagging DNA strands, right? That's what brings in the complementary adenine, thymine, cytosine, and guanine. And then DNA ligase joins the fragments together by forming phosphodiester bonds between nucleotides. So the DNA ligase is what seals the deal, essentially. So just to kind of put things in perspective, chromosomes are so small, but there are up to 80 million base pairs, so that adenine, thymine, cytosine, guanine, found in a chromosome. And DNA replication happens in small segments of the chromosome, because if it did not, in humans, DNA is copied at about 50 base pairs per second. So if we would do one whole chromosome at a time, it would take a month for DNA replication to happen for one chromosome rather than the hour it actually does without the multiple places on the chromosome where replication can begin. So remember, those DNA primers or those RNA primers make it easier for replication to begin in those certain areas, right? Those Okazaki fragments can be added, 2 new strands are made. So it's a really complex process, but it happens without us really even knowing it because it's so small and it's so microscopic.